Good afternoon, dear students. Today we are going to continue with the concept of gene. In the last lecture, we studied what is a gene, what are the different units of a gene, that is cistron, muton, and recon, and what are the variations that are found, that is overlapping genes or gene within gene. Now, let us discuss today about split genes. What are split genes? Okay. Now, during 1970s, in case of some mammalian viruses, for example, adenovirus, okay, it was found that DNA sequence which codes for polypeptide chain or the DNA sequence which code for a protein were not present continuously. Okay, what was found that in 1970 it was found that mammalian viruses, for example, in case of adenovirus, it was found that the DNA which codes for the DNA sequence which codes for protein or polypeptide was not a continuous sequence, but it was split into several pieces, right? So the DNA which codes for protein was not a continuous sequence, it was split into several pieces. Therefore, these genes were variously called as split genes or introns or interrupted genes or intervening sequences or inserts or junk DNA, right? Now, so where the existence of split gene was found first? In case of adenovirus, that is mammalian virus. Okay, what was found? The DNA which codes for protein is not a continuous gene, it is split into small pieces, right? And hence it was called as split genes or interrupted gene. And the interrupting sequences are called as introns or intervening sequences or junk DNA or inserts. And for the discovery of split genes in adenovirus and higher organisms, Roberts and Philip Sharp were awarded a Nobel Prize, right? Now the coding region containing actual information of the gene of most eukaryotic gene are interrupted by few to several non-coding sequences which are called as introns from the word intervening sequences, which are spliced out after transcription. Now let us understand this. See, in case of the you can say adenovirus, where it was found first, as well as in case of eukaryotic organisms, the eukaryotic genes are not continuous. That means the genes which code or which contain the information for the synthesis of protein, okay, are interrupted in between by few to several non-coding regions. Okay, so there is a gene, eukaryotic gene. There are regions in the eukaryotic gene which contain actual information to get transcribed and translated into protein. Okay, and these coding regions are interrupted by several non-coding regions. Okay, so these several non-coding regions are called as introns or intervening sequences, and the coding region which actually contain the information for the synthesis of a polypeptide or a protein are called as exons. Now the introns or the non-coding region present in between the exons are spliced out after transcription. Such genes which contain coding region interrupted by non-coding region, that means the genes contains coding region which codes for protein called as exons, which are interrupted in between by non-coding regions called introns. Okay, such genes which are split up into exons and introns are called as split genes, since their coding sequences are split into several parts due to the presence of introns in between. Okay, so if we draw a DNA like this, so this, for example, is a gene. Okay, so it is split up. Say for example, number 1 is exon, number 2 is exon, number 3 is exon, and number 4 is exon. Okay, But in between 1, 2, 3, 4, which I am calling as exons, which are the actual coding regions which are going to take part in protein synthesis, 1, 2, 3, 4, they are going to take part in protein synthesis. The information required for synthesis of protein is stored in the coding region 1, that is exon, coding region 2, exon 2, coding region 3, exon 3, and coding region 4, exon 4. But the coding regions which are going to code for protein, which contain the information for synthesis of protein, that is 1, 2, 3, 4, they are not continuous. They are split up. Why they are split up? Because in between the coding region 1 and 2, a non-coding region is present, which is called as intron, which is called as introns or intervening sequences. This one is also an intron. This one is also an intron. Okay. So in between the coding region, that is exon, some non-coding regions called introns or intervening sequences are present. Such genes which are not continuous are called as split genes. Is that clear? Okay. Now in prokaryotes, the coding sequences of genes are continuous. In prokaryotic organism, the genes are continuous. They are not interrupted. They are uninterrupted gene with a very few exceptions. All prokaryotes do not contain split genes. Their coding region is continuous. It is not split up. It is uninterrupted. Okay. So look at this diagram. You can see here we have shown a DNA sequence here. Okay. So this is exon 1. Okay. With the dark color we have shown intron and exon 2. That means exon 1 and exon 2 are coding regions. Right. Intron is the non-coding region. Intron does not contain any information required for synthesis of the protein product of this particular gene. Right. Now, so how to remove intron? The region which is non-coding, that is intron, is spliced out or removed during transcription. Okay. And then after translation, we can say a protein product is formed. So the exon during transcription is spliced out. You can say, sorry, the intron. The intron or the non-coding region, as you can see in the diagram, is spliced out during transcription. And what you get is a mRNA having length shorter than the DNA. The mRNA is having length shorter than the DNA. So as shown in the above diagram, you can see that the DNA sequence which code for mRNA, okay, the DNA sequence codes for mRNA, but the complete corresponding sequence of DNA is not found in mRNA. The complete corresponding sequence of DNA, that is the gene. Suppose I name this gene as gene 1. Okay, the complete gene 1 is not transcribed into mRNA. Why? Some region of gene is missing in the mRNA. The region of the gene which is missing in the mRNA is called as intervening sequence or introns. And the sequence of DNA which is transcribed to form the mRNA, they are known as exons. Okay. So those regions of the gene which form the mRNA, which are transcribed into mature mRNA, which is going to form the protein, are called as exons. And the non-coding regions which are not present in mature mRNA, which are not involved in protein synthesis, are called as introns or junk DNA, or you can say inserts, or and the gene is called as split gene or interrupted gene. Okay, you can see very well in this diagram now again. So this is a gene shown here. So there are exons shown by blue color and introns shown by red color. Okay, so most of the time what happens, the whole genome, the whole DNA gets transcribed into RNA. That means both actual. See, when split genes were first discovered, scientists tried to know how the transcription and translation or process, protein synthesis takes place from such a split gene. Okay, several hypotheses or several mechanisms were put forward. But finally, it was found that the whole gene, the whole DNA gets transcribed into RNA, right? That means all the introns as well as exons are transcribed to form the mRNA. But then post transcriptional modification take place. Okay, that means first, as you can see here, exon, intron, all have got transcribed to form mRNA, right? So transcription and all the DNA gets converted into mRNA. The whole length of DNA gets converted into mRNA. Introns as well as exons are transcribed, but then post translational modification. Okay, during post translational modification after transcription, during post translational modification, spliced out. What are spliced out? Some part of the gene mRNA is spliced out. What is that? The introns, those regions present in the mRNA which have transcribed from introns are spliced out specifically. We can see here in this diagram, the mRNA is formed. The red region which is shown here is formed from intron. The intron region is carefully spliced out to form a mature mRNA. The length of the mature mRNA is shorter than the length of the DNA. Why it is shorter? Because only exons are present in the mature mRNA. Regions transcribed from, I can say, exon is present in the mature mRNA, and only this is going to take part in protein synthesis. So amino acid chain, and after translation protein, the whole organized protein is formed with all secondary, tertiary structural, you can say, stability. Okay, so in this diagram makes it clear that this is a gene. Okay, it contains introns as well as exon. The whole gene gets transcribed to form RNA, right? After that, splicing out, that means post transcriptional modification takes place. What is spliced out? All the regions. This is intron, this one is intron. All the red ones which are introns are all spliced out. So splicing out of the intron region to form mature mRNA contain only the region transcribed from exon, and this takes part in protein synthesis. So that is about split genes. The mechanism of mRNA synthesis from a DNA containing introns or intervening sequences can be explained as RNA polymerases synthesizes both intron and exons. Okay, so RNA polymerase, when it is going to produce mRNA on the DNA, it will synthesize everything. It will transcribe everything, transcribe intron, exon, everything. So RNA polymerase synthesizes both, transcribes both introns and exons, and processing of the transcript occurs later on. Okay, so after transcription, the mRNA transcript formed is processed. The transcript that corresponds to introns are removed. Intron region is removed and exon region is left behind. The first report of split gene was provided by Hexon gene of adenovirus and albumin gene of chickens. So these are the two examples of split genes where split genes were first found. Hexon gene of adenovirus and albumin gene of chickens. Now let us discuss about Hexon gene of adenovirus. The DNA of adenovirus is double-stranded and codes for 20 proteins, right? Out of the 20 proteins, 8 out of the total genes are called as late genes. Okay, out of 20, 8 genes are called as late genes because they transcribe mRNA very late in the virus life. Okay, there are certain genes which are called as early genes which transcribe early. There are certain genes which are called as late genes because they transcribe late in the life cycle of the virus. The 8 late genes, okay, in case of adenovirus, the 8 late genes transcribe and form a single RNA which is then cut into 8 mRNAs. Okay, so all the 8 late genes of adenovirus get co-transcribed into a single RNA. Single RNA is formed, and then the single RNA is cut into 8 mRNAs. Okay, now the total length of the 8 mRNA is, however, less than the length of the original transcript. What I said? All the 8 genes get co-transcribed into to form the long mRNA, polycistronic mRNA. And then the mRNA is cut into 8 pieces. But when you look at the length of the 8 mRNA, it is shorter. The total length of the 8 mRNA is, however, less than the length of the original transcript, original long polycistronic messenger. Why? How? Now, when one of the genes, out of the 8, when one of the genes, that is Hexon gene, was paired with his mRNA, the DNA formed 3 loops which represent the unpaired regions. Try to understand this point. See, the total length of the 8 mRNA is shorter than the length of the original transcript. Okay? Now, when scientists wanted to see why the length of the mRNA is shorter than the length of the transcript, so what they did, they took one of the gene, that is Hexon gene, and the Hexon gene was paired with the Hexon mRNA. Right? When the Hexon mRNA, out of the 8, the Hexon one was taken, or Hexon mRNA was taken. So the Hexon gene was paired with the Hexon mRNA. And when they tried to pair the DNA with the mRNA, the actual thing is, because mRNA is formed from DNA, the DNA mRNA hybrid should be formed. Right? But then what they found, that when the Hexon gene was paired with its mRNA, the DNA formed 3 loops. Okay, that means the looped region of the DNA was not present in the mRNA. So, when Hexon gene was paired with its mRNA, the DNA formed 3 loops, telling us that the looped region is the unpaired region, which will not pair with the mRNA, whose part, the loop part of the DNA, that is not transcribed, or that is not present in the mRNA, and that loops were actually representing the introns, interrupted genes, which do not take part in protein synthesis. They were actually the introns, and the region of the Hexon DNA which paired with the mRNA was only the exon region. Okay, this indicated that Hexon mRNA was transcribed by 4 different regions, unlooped regions, and the 4 segments were then joined together. The looping is a means to exclude, there is a slight spelling mistake, the looping out of DNA is a means to exclude unwanted DNA which does not transcribe mRNA. That is, the looped region is the intron region, and the region which pairs with mRNA is the exon region. Okay, so you can look at this, the Hexon gene of adenovirus shown in black, that is DNA, Hexon gene of adenovirus. Okay, with its transcribed mRNA. The red color is the mRNA. So when the mRNA was paired with its parent gene, Hexon gene, you can found out that it was found out that 3 regions looped out. This is A loop, this is B loop, and this is C loop. So the black one is actually the DNA, the Hexon gene. Okay, and the red one is showing the mRNA. So you can see that the length of the mRNA is shorter than the length of the Hexon gene. Okay, so when you want to hybridize the gene with the mRNA, the DNA loops out and then forms a pairs with its mRNA. So the looped out region, what is the A, B and C loop representing? They are the introns, okay, which are not going to take part in protein synthesis. And the unlooped regions, okay, they are the exons which pair with the mRNA, and they are called as exons, and they are going to participate in protein synthesis. Similar observation was made in ova albumin gene of chickens. Okay, so P. Chambon's group and B.W. O'Malley group, they showed that ova albumin gene of chicken is not continuous, but it is made up of pieces scattered in the chromosome. Such genes were called as split genes. The structure of ova albumin gene in chicken was considered in detail, and it has been found that the split gene of ova albumin, okay, contains 8 exons and 7 introns. That means if you want to pair the mature mRNA of the ovalbumin gene with the ovalbumin gene. Okay, when the mRNA of the ovalbumin gene was paired with, if you pair it with the gene, you will found there will be 7 loops. Okay, what are the 7 loops? Those are the introns which do not participate in protein synthesis. Okay, the structure of ovalbumin gene in chicken has been studied in detail, and it was found that it is a split gene. Okay, and it has 8 exons and 7 introns. The size of the ovalbumin gene has been shown to contain 7,700 base pairs, although the mature mRNA contain only 1,872 nucleotides, right? When I say mRNA is formed from DNA, then the mRNA should also contain 7,700 complementary base pairs, but it is not the case. In ovalbumin gene, the mature mRNA contain only 1,872 nucleotides. That means the remaining are actually from introns. It has also been shown that what happens first, excuse, the entire ovalbumin gene containing 7,700 base pair is first transcribed into precursor mRNA. It is first transcribed. All 7,700 base pairs form a complementary mRNA with complementary bases into a precursor mRNA. The precursor mRNA then undergoes post-translational modification. So the mRNA transcript is modified. How it is modified? All the intron region which is not going to participate in protein synthesis loop out, and they are spliced and cut out. Okay, so all the intron region loops out and forms they are looped out and they are removed, and ultimately a mature mRNA is formed. So you can see here we have shown in the first, this is the first diagram which is showing you the ovalbumin gene containing 8 exons shown represented by A, B, C, D, E, F, okay, G, and the exon region. Sorry, A, B, C, D, E, F, G is representing the intron region, and the remaining is the exon region. So there are 7 introns. Okay, so whole ovalbumin gene gets transcribed into form a precursor RNA, and then what happens? The precursor mRNA is modified. You can see here post transcriptional modification is taking place. So these are the loops A, B, C, D, E, F, G. Okay, so these are the 7 loops. The 7 loops are of 7 introns, and all the loops are removed by splicing. So you can see all the loops A, B, C, D, E, F, G, they are removed by splicing. The precursor mRNA is modified, and what you get here is the mature mRNA. What is this? It is a mature mRNA. Okay, only the mature mRNA will take part in protein synthesis. So you can see that the length of the mature mRNA is shorter than the length of the DNA. Okay, so this is how transcription of ovalbumin gene of chicken take place. Now if you look at this diagram, you can see A, B, C, D, E, F, G all have looped out. What are the looped out regions representing? They are representing the introns, and the unlooped region shown by 1, 2, 3, 4, 5, 6, 7, 8 they are representing the exons. Okay, and the exons pair, the mRNA formed from the exon will then produce the protein. Okay. Now the split genes also have been found in beta globin gene of mice and rabbits, tRNA genes of yeast, and ribosomal genes of Drosophila. And existence of split genes is very common, or it is found mostly in eukaryotic organism. But prokaryotic organism, almost all prokaryotes do not contain split genes. Their genes are continuous. Okay, so that is all about split genes. And now here, after this, we are going to discuss what is a polycistronic mRNA and what is a monocistronic mRNA.